I'd like to welcome our audience to our live webinar, Is Your Drug Discovery Ready for AI? A maturity model for GenAI in drug discovery. My name is Toby Liebsch. I'm a growth lead here at Cradle in Amsterdam, and I'll be your host for today. With that, I want to welcome today's speaker, Cradle co-founder and CEO, Stefan Grieken. Hi, Stef. Where do I find you today?
I'm in Zurich. Good to see you.
Wonderful. And with that out of the way, Steph, I'll hand it over to you.
Excellent. Thanks everyone for coming. I'm excited to be talking a little bit about how to think about maturity of AI in biologics discovery and development. This really came about as a lot of our clients were asking, hey, how should I think about integrating this into my day-to-day workflows, right? There's sort of all kinds of cool models out there. There's a lot of research out there, but how do I make this standard of care? And so today we'd love to share some of our thoughts and then based on your questions, some reflections on how to make your organization AI ready. First, briefly about Cradle. We're a software tool that you can use to engineer proteins. We fit seamlessly into your existing workflow where you design your large libraries or your plate-based libraries on the platform. You still run your own assays, and then you can bring data back to Cradle. It's a web application as well as an API, and then obviously a lot of this is built on top of our foundational model that we give scientists access through. Some of the things today will all be referring to our own in-house lab. We have a lab that we use for two reasons. One, think of it a little bit as our self-driving car, where we try to A/B test new ML methods on a wide range of enzymes, therapeutic proteins of different sorts, and different ranges of assays, across initial hit identification, all the way down to sort of late functional assays. And then the second thing we do there is we develop data sets, and sort of as we have built that lab, we've also learned a few things about how do you get ready, because we're a customer of our own software, if you will. We currently have 25 customers on the platform, over 30, I think it was 40 something when I last looked, programs that are active. And so, we're seeing at this point quite a good proportion of I think representative proportion of programs across the industry. Okay. So let's start first about sort of what's really the promise of GenAI, especially as you think about it in the drug development process. And I'll take the drug development angle. I know there's a couple people out there that are coming more from the industrial biotech side of the house. I think the answers are the same, just the words are somewhat different. So we had a few questions from the audience. One was, how do we educate the rest of the organization about the value of AI? And then how do we create a little bit more urgency and speed around adoption? Thanks, Jorge, Beth, and Magnus for these questions. I'll try to sort of contextualize it a little bit with sort of how we think about it here. So, obviously it's a pretty long process to get from sort of your initial validated target all the way down to, let's say, a fully launched product. And, I think in contrast to maybe other industries like maybe law, where you can use sort of the same model for many different tasks, actually, the process of coming up with a molecule is really something that, requires different types of machine learning models and different types of expertise across the different processes. But in the early research space, it's really organizations like Future House that are doing a really cool work where you could maybe scan through large corpuses of literature and get that in a highly reliable way, curated and summarized. You might do some hypothesis generation and validation using a language model. And there's a bunch of companies out there that are doing some really cool work around large genomics data sets or real world evidence data sets that are out there, really with the goal of ultimately coming up with some understanding of a mechanism and some hypotheses about how to validate that mechanism. Now, where Cradle then typically comes in is once you've decided this is my set of targets or the molecule and the type of molecule that I want to develop in order to potentially come up with a cure. Here, the models really change, right? They start working, much more on sequence data in our case. You generate molecules, and then you go through lead optimization to preclinical. In the earlier stages, I think you'll discover more targets. You'll have a much deeper understanding of your target biology, and you can probably get to quicker discovery. When it comes to Cradle, really how typically the stages from initial hit ID to manufacturing people judge us is, can you get there in fewer experimental rounds? So labs are absolutely still in the loop, but do you just need fewer rounds to get there? Second, and I think this is really where some of the de novo methods shine, can you get to targets that were harder to target? And then thirdly, can you really get to better molecules? So can you get to things that are better binders, sort of better developable right from the start when you're doing hit ID? In lead optimization, ultimately, can you get there as well in fewer rounds or with fewer data points? Can you get to better TPPs? And so here, multi-property optimization, where you want to have something that's stable, that binds well, that is potent, that expresses well, et cetera, et cetera. And a lot of these things, contradict each other. And then lastly, and this is starting to become more relevant, is can we translate more towards, let's say, later stages in clinical studies? So, can you, for example, engineer out immunogenicity more easily than you could with some of the methods that exist there? Translation is still complex, right? And I don't want to claim that anyone has solved that, but I do think you can evaluate virtually a lot more compounds for certain liabilities that were, let's say, harder to do if you were to rationally design them or work in a more combinatorial way. I think in those early stages, higher volume of programs, speed and complexity and quality is really what you drive. And then that should ultimately lead to a higher probability of success in clinic. And so how do we think about it? When you think about sort of new capabilities, both efficacy and safety, and so you can get to higher quality molecules. And if you want to have more interesting things into your portfolio, such as intracellular targets or GPCRs, those should become easier to target with some of these methods. Then, and a lot of the managers will enjoy this, getting there with fewer time spent and fewer costs spent, and then hopefully reducing risk in the long term, getting to potentially a best in class or having a slightly higher success rate in clinic. That should, at the end of the day, yield a higher return on R&D investment. Now, when leaders or managers ask me, hey, why should I invest in these types of programs? It's really the obvious thing, right? It's quite expensive to run these things. And so this is a survey that we've done under some of our cost base and then, client base. And you can see the cost base for, a lot of these programs is quite high, where, a significant amount of people spend months or quarters to years in some of these phases. And they will typically spend more than 500,000 every month on, let's say, lead optimization as well. And so, how do you compress the time that you need, and how can you make the cost lower? So we're seeing sort of three approaches right now, and sometimes companies will combine them in how people are trying to bring AI to biologics. And so you can make and build in-house. So here, typically, you would set up a team. The successful teams are roughly around 40 to 80 people. I think a good example is Prescient Design at Genentech, and they're building in-house software that can support many different biologics development programs. Then the second category is companies that have certain AI capabilities and are more asset focused, and so take a Perfluent or an Inceptive or Isomorphic Labs. It's more from a business perspective very similar to how you would engage with another asset-oriented company, but it's it's still sort of you work on a set of targets or a set of programs together. And then thirdly, you can buy software from vendors like ourselves and get it into the world. Now, a lot of pharma companies and biotechs ask me sort of how do you get ready for AI and sort of leverage really the full benefit of it. And this is our first attempt at coming up with some sort of maturity model for the organization, everything around the actual molecule generation. We'd love to get your feedback on this as well as we go through it. So feel free to send me email, or at the end we can discuss it a little bit. So let's first start at capabilities, because I think that's an important thing. So what can you do with AI and what can you not do? So here we had a few audience questions. One is, how do you build trust in these AI models and predictions? And how do you establish validation for these applications? Great questions, and we'll dig into that. Second is, how do you make in silico, so any generated variant that you have, correlate well to what's going to actually happen into the lab? That was a question from Magdalena, and we'll try to answer that. And then finally, translation of ML findings into in vivo, a question from Emma. Let me try to answer that last one first. That is still very hard. I don't think AI models are at a point yet where you could say, this is my mechanism, engineer me a molecule, and I have a very high guarantee that that translation will happen in vivo. It's mostly because of the complexity of the organisms that we're trying to measure the effect of, and those systems are just way more complex than, let's say, two proteins interacting with each other or a protein interacting with a cell. We might get there, but today that is still fantasy. I do think there are proxies for translation. For example, will something bind to MHC2 and cause some sort of unwanted immune response? Those are things that are getting more possible, but that's still somewhat of the frontier. I do think these other two are definitely things that you can solve, and I'll give some examples of those. So when I look at organizations, and we obviously talk to many, we have many customers ourselves, we can sort of see a trajectory from, let's say, initial human assessment all the way to computers being really in charge of figuring out what sequences get tested. So human assessment, this would be machine learning really serving only to supplement human knowledge and help human assessment of a candidate sequence. So here, you might run a structure through AlphaFold and decide on certain mutations in a more rational fashion. You might use a PFAM label to get natural enzymes, starting points, that kind of stuff. But it's really used as an aid for people to then do what they were doing before. The middle category, and I think most companies that do build in-house are sort of in that category. This is where machine learning basically serves to be a judge, right? It's scoring and aiding of selection of candidates, and then trying to provide some form of holistic assessment of a candidate. And so here it could be things like, hey, the immunogenicity scores I just talked about, is this going to be highly or not very highly immunogenic? Any sort of folding, docking, binding heuristics you might get from a model, and then ultimately scoring things on, is this diverse, et cetera, et cetera. Then the third category is where machine learning doesn't just judge, but it also generates. So it's both the creator and the judge, and then both of these get sort of steered by humans as they try to develop their candidates. So here, sequences that get tested together, you're generating plates, right? A set of outcomes that you're looking for. They produce variants both without experimental data as well as where there is experimental data. There's some understanding of confidence, right? And this was in some of the questions from the audience as well, where you have some understanding of, will this translate into my wet lab experiment? And then selection and design of the experimental round is more in the hands of the model as well. So it's not just, here's the top 96. No, we're going to really pick not just for the top 96, but we're going to try to avoid risk. We're going to put up a few hypotheses in for what might happen around after. Here I'll talk a little bit about sort of how we see capabilities evolve, especially in therapeutic organizations. And typically what we see is that a team that's building in-house, or even if they're adopting software like Cradle, is they'll start on a single modality first, right? So it might be the single chain antibody team or whatever. And I do think when you're a large organization, you need to typically serve many different teams, and actually a lot of the open source models that are out there are often very single purpose, right? They're very much designed to do one thing well, and they might not cover peptides because peptides have non-naturals, or bispecifics because you can't optimize those bispecifics, or maybe not enzymes because it's really an antibody focused model. And so what we're seeing is over time, and this has actually been our journey as well, you move from a single maybe a single chain antibody to ever more different modalities being let's say available to scientists to use when you're building these software platforms. Second is sort of across tasks and phases. Right here, there's a lot out there, right? Do you use it only in target ID? Do you use it in lead op? Do you use it in repurposing, in formulation? And it's a little bit to my earlier point. In biology, the tasks are so different that there's typically different providers and different models that work well for a specific phase that you might be trying to support. Then the application needs to be able to support a pretty broad range of functions and assays that are required to really get to a fully developed molecule, right? So typically what we see is that customers will maybe start with some single property, right? It might be structure, or maybe they have an expression predictor or some other developability property. But typically what you would see is you would start from maybe a single property, go towards modeling multiple properties, but maybe not even just understanding the relationships between these properties. And I think this is really where we found a lot of value. If models understand, if I change stability in this way, how does it influence expression? And a lot of what you're trying to do as you're getting to a good TPP is making lots of trade-offs, and obviously a lot of these things have relationships to each other. Then reliability. This is, for those that follow me on LinkedIn, something that I've been harping on. When you see a new release of, let's say, ChatGPT, that's a multi-thousand person engineering team and a even larger quality evaluation team that comes up with a machine learning model. Now, in biology, a lot of the models that are coming out are coming from academia. They're respected models, but they're really demos, right? So they're one or two postdocs or PhDs that have worked on a certain model. And the breadth of application of these models is not always known. And especially given how expensive our job is, right? If a ChatGPT lies to you, it's fine. If you generate a plate of protein variants and you take them to your lab and they all turn out to be duds, that's not something that people like. And especially for us as a software provider, as should it be for any internal team developing these tools, you want to make sure it reliably works, right? Because otherwise, a lot of your customers, the scientists, are going to be like, hey, it didn't work in my application, and that really discredits your effort. This is a good example, I think, from a recent in vitro competition from the Adaptive, which is a CRO here in Switzerland. They effectively evaluated different types of methods that people were using to come up with a better binder against EGFR. What you find here, and I think for me the most interesting thing is there's a lot of people that are using, for example, the exact same de novo model or de novo pipeline, and it takes, and you get like dramatically different readouts for literally the same model against the same target, depending on the person that operates it. So reliability and robustness of these techniques is really something you need to take into account as you're building a platform, and that means evaluating, that means having benchmarking available. Only in silico benchmarking is not something you can do in our industry. We have a little t-shirt for our lab in Amsterdam that says in vitro or it didn't happen, right? You can't build a self-driving car company without driving in a car once in a while. Now, I think, and there were a couple of questions about how do you know whether you're going to get good results or not? And I think here, the big issue in biology is that we have relatively sparse data, right? If you take, I don't know, a ChatGPT, you can sort of download the whole internet. You get a pretty decent coverage of things humans will typically say, and you can sort of throw more data at it. We don't have that benefit in biology, right? There's very limited sequence data just available based on what we've been able to sequence from natural diversity. That is probably a small subset of what's plausible with biology, but there was never an evolutionary pressure to get there. And then of those raw sequences, we don't have a lot of readouts. We just don't know what they do. We don't know if they're stable. We don't know if they express well, et cetera, et cetera. And so that means that our models in our domain, within protein engineering will go out of domain a lot faster than many models that are out there. And so you need ways for your models to introspect and say, hey, I'm in domain or not, or I can make, let's say, a certain edit with a certain amount of confidence. And so this is actually a picture of sort of a very long campaign. This was an enzyme project where the only information that was given to models was round one, and we were trying to predict readout values as you're moving towards incremental rounds of optimization. And you can clearly see the drop off here of your predictive value. Now, around round 9, models should have been saying, right, this is where we start. This is sort of roughly where we can still try to come up with good results. But beyond that, you're a little bit on your own. To give you an example of how we do this in Cradle and how we built that trust ahead of a round, is we do two things. One is we try to explain in the eye of the models what might happen. And so this is 2 template sequences, which are the dotted lines, and what the model thinks the outcomes might be in terms of lab at a certain confidence interval. So and this really helps people in silico to then still say, hey, I don't like this 96 well plate. I'm going to go back. I'm going to change some parameterization. I might do more mutations, or I might alleviate a certain constraint, or make other changes that will potentially give me a better outcome in terms of what might happen in the lab. We're currently even capable of doing that at, hey, we can, for the top 10, guarantee that you'll have readouts within this range, which is very helpful. And if you do that consistently, it's a little bit like how Google Maps tells you it's going to take 2 hours. If it actually takes 2 hours, then you're happy. And if it does that consistently, you'll trust it as a tool. I think the second thing, and this is a little bit to sort of the third part, like, can you generate not just or score not just a variant, but can you build out a plate or build out a large library that that might hit your general objectives? You can actually, if you try to design libraries or plates, you can actually play a little bit with things like, how confident are we here? Can we maybe take some risk here to learn more for my next round? Can we definitely have a few good binders that my management will be happy with? And so effectively trading off sort of some of the risk reward that you have as you build out that plate. That will give you consistency over time. And again, scientists that get tools that work consistently, that work well, and they work as they say they will, is something that we found is quite important for people to retain and continue to use the software. All right, let's get to the favorite topic, data. Data is a real issue, right? It's complex and it's hard to organize. So many people ask questions about that. How do you address data quality issues? How do you do training data curation, bias detection, noise reduction in biological data sets? So I'll talk a lot about how do you capture data quality issues, training data curation, bias detection, and noise reduction. We ended up deciding we need models to be able to deal with those things. And so for Cradle, there's not a lot of, all of those things are automatic and encapsulated in the ways our models work, because it is noisy, right? By definition. And you're not going to get around that, and sort of humanly curating every potential data set that comes out is hard, right? We found customers where there were like environmental effects in their labs on Wednesday, where certain temperatures became a little bit too high that changed things. You have plate effects, you have edge effects. Effectively, your models just need to be robust against certain quality problems. That said, there's a lot you can do to make sure that you get the highest possible quality data coming back from your lab. And then what's the right data structure for AI? That's a great question. We'll dig into that a little bit more as well. So the first thing that we're seeing is, you can obviously collect data for the purpose of developing a product, like an individual molecule against a certain therapeutic target. But actually, a lot of the work that we do at Cradle and something that our customers pay for is just literally evaluating ML methods, right? And so there, you don't need, let's say, a single project that you try it on. You need some form of benchmark that you can do continuously, right? And so, when I was still working at Google, there was a large team in search called Search Quality, and their whole job all day was to figure out how do we make the quality of those models better. In biology, that means you have to go into the lab. And so, every 2 weeks we run, rounds of assays where we test out new techniques on, specific proteins. And that helps the machinery of getting better at making this software, because at the end of the day, this is a software development process, right? If you don't do this, you're building a self-driving car without cars, and that is a very complicated thing to do. Second is assay quality. Assay quality varies a lot. And the best thing that we found is to provide feedback to scientists and make models more robust against these types of batch effects. So what you're seeing on the right-hand side here, every bar is the same assay measurement on the same sequence, and the box plot basically gives you the range of outcomes there. And it's obviously really hard for a model to deconvolve what's better here than anything else. One, give that feedback to scientists, and it can come in many different ways. You can show it to them like this and have models say, hey, you're out of the norm, right? This is quite noisy. Are there ways in which you can improve that? And that's obviously something a scientist will have to work on. You can do things like, we're detecting often, hey, your assay is like hitting his dynamic range, right? Like you can't measure below this thing, but you've set that as an objective. So maybe you want to go back to your assay and see if you can improve it. And then at time of acquisition, so when you get the data in, there's a lot you can do to basically avoid people uploading low quality data. That said, the nature of biological experimentation is that data is sometimes noisy, and really what you can do is more guide, replicates. You can do those types of things, but at the end of the day, your models need to be robust against noisy data, because otherwise you're just not going to succeed. Second is throughput. What we found is 96 is typically fine, but you're not going to get a lot of value if you're dramatically lower than that. Now, this is mostly because models need to be able to build confidence, right? And they need to hedge their bets a little bit. So getting to 96 at a minimum is what we recommend. We are seeing customers that may oversample a little bit. So, that have throughputs of like, 384 plates or, around 1,000 samples. That seems a little bit like overkill and is also quite expensive to do. And so, if you're in that category, AI can really help you to lower your cost per sample. Synthesis techniques are quite important. Now, for some pharmas, nobody cares, right? You can just buy linear sequences from Twist. But especially in industrial applications, sort of control over the sequence is not always guaranteed. And so I think one of the things that we found is, especially with AI, you need a lot of control over your synthesis methods. And you need to be able to make edits that are pretty far away from each other, effectively anywhere. We love gene fragments and assembling them in-house. There's another talk done by my co-founder Harman at Gen. I think there's a webinar, we'll put it in the notes, where he talks a little bit more about how we do DNA build at Cradle. Latency. Cycle times really matter. One of the first things, and I'm not a biologist, I was on the product leadership team of Google Research before starting Cradle. But one of the things I did always know from back there is the faster you can make the feedback cycle to the machine learning person, the better the models will get ultimately. And so on average, we are seeing 10 to 11 weeks of turnaround time for an experimental round between sequences ready and experimental results coming back. For most ML researchers, they've kind of forgotten what they were working on 11 weeks ago. And so if there are ways in which you can reduce your cycle times, that significantly increases learning. And the additional benefit is that your bosses get more happy because your lab just gets you to where you want to be a lot faster. We've currently brought it down to 2 weeks on average, and we think there's even still a lot of headroom to go down further. I'm still hoping for Amazon next day for antibody development at some point, but talk to me if you know how to get there. Then automation and integration. The more you can have, especially tasks that require a lot of human attention or that are error prone, if you can let that run by computers, so not have people copy pasting data from A to B, not have people have to put readouts into a spreadsheet where they might put the comma in the wrong place. But also things like, if they're liquid handling by hand, those are things that are error prone. Humans are, in theory, good at it, but it's just not the most interesting work for them. And we found the more you can automate these things and make them really user-friendly for people, the better. I have a little video on how we do sample normalization at Cradle. And I think, Toby, you're going to play this video, right?
Yes, that is correct. Let me play the video here, and it's shown up inside the Cradle lab. Seems to be a technical issue right now. Let me see what I can do. So
play here.
You see it plays live here. There seems to be a technical issue
Hi,
at this point.
I'm Dan,
Let me
Research
see.
Associate at Cradle. Cradle is a platform that helps scientists engineer better proteins faster with AI. But we're not just a software company. Our Amsterdam wet lab validates models and feeds them new data. Biology has slow feedback loops. The faster we can generate lab data, the faster we can improve our AI models. That's why we use automation across our lab workflows. Today I'll show you an automated workflow that normalizes sample based on protein concentration and also discards low-quality samples, all within a few clicks in Benchling, our LIMS. Normalization is essential for reliable protein assays, so you often need consistent concentrations. We build a system that handles this automatically and tracks everything digitally. This means that we avoid carrying around USB sticks and spending hours filling out data sheets. This makes the process simpler for scientists, which means fewer mistakes and therefore better data quality. For this workflow, we use two instruments: the Tecan Spark Plate Reader, or BCA assays, which measures the protein concentration, and the lab chip for protein purity measurements. When instruments create measurement files, our parsing workflow automatically puts clean data in our data warehouse. Now, let's see how we analyze and normalize the data in Benchling. All data flows into Benchling through connected apps. These apps pull from our data warehouse. This is embedded within our Benchling canvas, which means both the automation and documentation happens on one page. We can now fill in the protein quality control canvas, which takes in the data from the Tecan Spark and lab chip in combination with the quality control threshold. When running this protein QC, the purity and concentrations are calculated in the background with information from the data warehouse. In the meantime, visualizations are fed into the documentation, which helps the scientist to make a decision to continue or not. The commands for the liquid handler to go from the protein plate to the normalized protein plate are also calculated. These graphs show good data quality but reveal a plate effect. There's an uneven distribution in quality control passes across the plates. To continue our work, we need to normalize. Using a bidirectional connector, we can prepare the Tecan Fluent liquid handler directly from Benchling. The deck is prepared, and after that, the normalization begins. This automation delivers 4 key benefits: automatic data handling, a centralized workflow, scalability, and a reduction in human error. Combined with our other lab automation, we've moved from months-long turnaround times to a consistent 3-week cycle. We generate assay results much faster with all tedious work handled automatically. We will continue adding instruments and expanding our automation capabilities. Each addition gives us more data types to work with and helps us refine our models and test whether our protein designs actually perform as expected in the real world.
And if you're ever in Amsterdam, do let us know, because you can visit our lab. We also don't deem our lab methods proprietary, and so we're happy to share protocols and other things. Some of them are on our website as well. Great. Storage is a real issue as well. So, many of our customers have LIMS systems, but there's a lot that gets lost. So, for example, you saw some of the work that Dan was just doing in our lab. We try to, for example, get the original measurement data, so SPR curves for binding assays and those types of things, because there's actually a lot that you can learn from that data. The most modern organizations that we work with will have some sort of data lakes. There's like identifiers across every experiment. There's clear, role-based access for all these things, and it's not hard, to find data. Unfortunately, there is still a bunch of customers like, for example, the one at the top where most preclinical data was really spreadsheets all the way down. And in those cases, we need to sort of provide those capabilities to the organization. So if you were to use Cradle, there's a couple of things that we provide. So one, our customer success team will work with your lab to set up schemas for all of your instruments and the ways that you collect data if you haven't done that already, so that it's easy to import data into Cradle. Second is we do a lot of data quality checks at ingress. And so, we catch errors in this is not a real sequence, this measurement is wrong. There is probably an effect here that you need to deal with. And then before we, quote unquote, validate and allow that data to be used for training, the scientist who has the real knowledge of the actual assay can help provide feedback and potentially discard certain data points, or at least mark them as suspicious, which is helpful as we're training models downstream, not downstairs. And then finally, scalable access. So there's definitely things you can learn between projects, and you need to think about how can projects leverage each other's data in the correct way. So for example, an expression host might be used for the same antibody or for different antibody projects at the same time. You can definitely learn things from these other projects. And so in Cradle, for example, we allow teams to say, hey, things that are common may be used by other projects, and we can transfer learn between these different projects. Cool. Finally is organization, and sort of how do you organize for AI? And here there's a lot of really interesting things. I think many companies get stuck here, and it's for different types of reasons. So a couple of questions. So wet lab and dry lab collaboration, how do you get this interaction between the two? Second is, how do you change that culture and that human factor, which is a great question. And what do I need to get started? I guess Julian will get an email from our team soon. Coming from AI, and there's a lot of hot air out there. There are a lot of senior leaders that are chasing hype. So there's not really a strategy around what do we want to use this for? What are the success metrics? How do we bring something from, let's say, an initial experiment where we do some sort of POC into something that's used by everybody? There is a lack of investment in enabling technology, so there's lots of like shiny objects that get bought, but the actual plumbing is not there. And then, depending on the organization, the amount of literacy within senior layers is not always there. And so, really, it starts with, for example, a leader saying, "Hey, we want to take this seriously. We want to get to a point where we embed it in every use case. I'm going to measure it in throughput, in the quality of the molecules, and that." And this is what you need to show me for this to be interesting, instead of we have some chatbot thing that we deployed to the entire organization. This one's been a pretty big one. For some of the companies that we've worked with a bit longer, we're starting to see ways of working change somewhat. And in some cases, this might actually change team composition. And so the, let's say, most classical thing that we sometimes see is there's different teams for different stages. So you might have a team responsible for hit ID, you have a team that's doing affinity maturation, the developability team, and then sort of more functional downstream teams. Really what we're seeing is sort of everything converging towards two steps. So step one, screening; step two, plates, done, right? And the more things you can run together, the better, which sometimes really creates attention and requires people to work together in different ways. I think the second one, which is more to the question on how do wet lab and dry lab folks work together, we really try to bring folks with the most knowledge of the system, of the molecule and the disease, into the process. So the whole goal of Cradle is to eliminate the AI expert as much as we can, because there's lots of things these models don't know about, right? And ultimately, it's a design process where both work together. So about the constraints, about downstream effects, you might know about structural features you don't want, you might know about your synthesis methods and all kinds of other things that no protein model could know, but you need to be able to encode and provide those priors to the models. There was actually an article in Biopharma Voice today where Novo Nordisk commented on this. Like, we've worked with them for quite a while now, and actually that was one of the big benefits for them, bringing wet lab and dry lab scientists together and engineering these molecules together, because neither of them can do this independently. Maturity is super important. So pharma seems to have a really good knack for doing POCs all the way down. But how do you mature, right? Sort of how do you get from running a thing, demonstrating it works, into something that's broadly adopted by many groups and many more people get the benefit of it? This is a very common practice in software development, right? At Google, you had what they call dog fooding. So you try your software first with your own team. Then there was typically a private alpha where there was a few early adopters that would sort of knock out all the edge cases. And then there was some way to ramp and deploy to many people at the same time. These processes are very often missing when it comes to AI use within R&D organizations. How do you deliver this is quite important. And so, what we see most often is a scientist trying some open source and applying it, but that is not something that's repeatable. If that scientist leaves, you're in trouble. We forgot the Jupyter notebook, right? That kind of stuff. Sometimes we then see folks trying to build internal tooling and workflows around it, so there's at least some repeatability. And then we think that at some point you need to get to something that's a little bit more enterprise grade, right? It's for me as a software person sometimes surprising that people buy Microsoft Teams to be able to send emails to each other, but a lot of folks want to roll their own AI platforms and molecular engineering tools, which seems like a lot of work to do. We'd love your feedback and understand a little bit more about what resonated with you, what we may have missed. But these are some of the general trends that we're seeing. Thank you for your attendance. And I think we're going to now go to the Q&A directly.
Fantastic. Thank you. Stef, indeed. Like mentioned in the slide just now, cradle.bio/maturity, you can fill out our type form to understand, give us a feedback on the session and your ideas on maturity, the challenges you see in your own organization, etc. I want to encourage you, if you have more questions, to pop them into our chat Q&A now. I see so far one question from the chat itself. I'm going to put it on the screen here. From Florian Schmitzberger, would you be able to comment on how you incorporate immunogenicity? And oops, that timed out quite fast. Would you be able to comment on how you incorporate immunogenicity, risk prediction in humans patients? I guess that goes a little bit back to our in vivo question earlier. I'm asking since data for molecules is hard to come by.
Yep, definitely. I think that's a great question. So there's different things that you can do for immunogenicity. There's more classical tools. So, there's known peptides that we automatically, peptide motifs that we automatically scan for that are being avoided at generation time. There are predictors. So for example, net MHC bind, we will take that into the generation loop. So we'll produce a billion variants. We'll look at scores for different allele groups, and then we'll be like, okay, we probably want to avoid this type of thing and try to avoid it that way. There is optimizing with assays. So there's a couple of vendors that we work with where you can run T-cell activation assays and optimize it out like how you run, let's say, your typical binding or SBR type of assay. That could be a way to try to avoid ADAs as well. And then finally, we as Cradle produce a lot of data in-house, in particular around peptide motifs that are likely to bind to MHC2. Now, that is not a 100% translation to you'll never get immunogenicity. You'll never get some form of anti-drug antibody formation, but at least it does limit the risk more than maybe some of the more classical methods.
Flora, I hope that answers your question. We have another question from Kelly. Thanks, Kelly. What is the biggest reason for R&D labs to be hesitant to integrate AI into discovery workflows?
It depends on the organization, but I think the biggest one is the opportunity cost. So, you're ultimately going to get promoted by the success of your programs. Let's say if I were to come into your organization and be like, hey, I'm Stef, we're going to go engineer all of your proteins that go into your next round of experiments, that will be tricky to do, right? And so, that would be risky. Spend 8 to 12 weeks of additional lab tests, tens of thousands, sometimes a lot more money, and people don't want that. Second, I do think some folks, they really want to understand why this works or doesn't work, and whether it will work for their specific use case. So let's say the burden of proof, like anything in science, is sort of on the person that proposes the method. And I do think that proof is building. I think 2 years ago, when I would talk to people about generating, plates of variants, they'd be like, "You're smoking crack." I think it's much more accepted now, but it's there's a cultural element to it as well.
Great. I'm going to also bring in some questions from that we didn't answer or maybe touched upon yet. There was a couple of questions about cost. Of course, we just addressed, so Damian, for instance, asked that question. We just addressed the opportunities and the opportunity costs of AI. But of course, many of these solutions also cost money. So maybe you can share some anecdotes here, Steph, from your conversations when it comes to actually selling Cradle and selling our solution and the considerations about costs that pharma leaders bring up with you.
Sorry, I don't fully understand the question. The question is, how do they reduce cost?
No, not how to reduce cost, but how like the solution costs, so the cost of the actual AI solution. Also, they're not actually they don't come for free. They also have to pay for them, and you have to navigate the opportunity cost versus the actual cost of paying for the solution for the software, with sometimes vague impact, at least perceived vague impact.
Yeah, I mean, so there's a few things, and this is typically what we would do with customers. So one, you need to do a proper business case, right? So let's say if you take typically 3 rounds of lead opt and 2 rounds of hit ID, at a minimum, we should be able to cut out a few of those rounds in order to make that investment worthwhile. So that's one. We've been, for the cases where we've worked with customers, always been net positive to, we've always been able to remove specific rounds and rounds of experiments from people's programs to date. Second, and that's a little bit harder to calculate, is can you get to a better molecule? This is an argument that you will have to have with your boss, but if you're in a very crowded space, for example, it might be beneficial to have a more potent, a more easy to manufacture whatever molecule that you're working on, but that's very specific to your organization. I think the thing that's mostly underestimated is building a platform like this in-house. That will require a bunch of researchers, a bunch of ML engineers, a bunch of people that can build application software, make it safe and secure and that kind of stuff. And I'm seeing a lot of the sort of people are trying to do the in-house thing. Then you get 2 ML researchers and no experimental bandwidth to try something out and improve on your methods. And then you'll be stuck in demo land for a while, is my sense.
That makes sense. Florian had a short follow-up question, which basically boils down to if you have something like T-cell activation assays that take months, how does that fit into the short cycle, and how do you solve for that?
Sorry, I didn't claim that we run 3-week turnaround time T-cell activation assays. That said, you should talk to Twist. So Twist has assays that take dramatically shorter and we believe to have decent translation. And so, if you believe that is a risk, you can try to engineer it out that way.
Yeah, we're also working on a webinar together with Twist. It's going to probably happen end of October on exactly that, how we work with them, our lab process. Our co-founders, Harman and Elisa, are going to present that workflow there, Florian. So that might be good to follow to get some more detail on that. I have a question.
Moving quite fast as well. There's significantly better assays coming onto the market, especially even since I started Cradle, I've been super impressed by all of the cool analytical chemistry methods people keep churning out.
Yeah. I'm getting some questions on the availability of the recording. Yes, the recording will be made available online. And I have 2 more questions here, and then we also have to close it soon. Can you comment on low data regimes? So you went into this a little bit earlier. Many R&D processes will not reach the throughput you find optimal, which I know is a question that we ask very much in the first conversations with potential customers.
Yeah, so it's absolutely true that if you're, for example, in academic labs, we still often have to say no for now because we find the data regime to be so restrictive, let's say, 10 or 20 samples, that we can't really help that much. It's the same, for example, we typically stop at your cell-based or functional assay. We don't go all the way in vivo if you're doing some animal studies on some limited number of mice. Like we couldn't do 10 mice, absorption stuff right now. And so we do find most, let's say, mid-stage plus large companies, whether they're in biopharma or non-biopharma, are capable of producing a 96-well plate.
Fantastic. And then I have another question from Kelly. Compliance and regulation playing into decisions to bring in R&D even in non-regulated labs.
Yeah, that's a really great question. So luckily for any application that we work on, you'll have to go through a proper clinical process. And so all the same rules for those processes apply for us as well as for others. That said, we are seeing, especially the FDA is quite excited about, in silico methods. I'm not fully convinced of all the translation ability, but I would expect in the next couple of years there will be more evidence of those techniques translating, for certain levels of confidence. And so I do expect translation over time and translation evidence will build, but it's just the nature of our business that we'll need to wait a little bit longer. In terms of other regulations, there's not that much different here, right? Like I think we've done a pretty good job in biotech, sort of regulating what kinds of sequences that you can make. What's somewhat tricky, so when we work on, let's say, de novo binder projects with against like targets that are much less known, that obviously carries a whole bunch of existing known risks, but it's more the nature of those targets being underexplored. So you might get mechanism wrong, but there's other things like, we don't know if things that don't come from natural repertoires will have different clearance. We don't know if they show up differently in terms of their immunogenicity. There's no reason. There's potentially more risk of off-target effects, right? Because you haven't evolved that binder through some natural means. So that's tricky, right? That's hard to say, and so we position those as more risky projects, and evidence will build, and we'll learn more about where those risks really are. But those are some potential risks there, and I could imagine, depending on how things go in the next few years, that an FDA or an EMA might put a different regulatory requirement in terms of clinical studies on molecules that were produced that way.
Thank you. And with that, Steph, there's one more question, which is a nice closing remark here as well from Lucas on the future of AI capabilities. Where do you see the AI model capabilities go in the near future?
Yeah, it's a really great question. So what's cool is that there's a lot more people that are building models these days. And so in the literature, you're seeing a lot of good stuff happening. David Baker had his big party a couple of weeks ago, and felt there was not a lot to announce there. So we'll see on structural methods. There have been a few cool papers by some of the companies that are out there, especially for smaller formats like mini proteins and peptides and VHHs. My sense is honestly that data is going to be the bigger factor here, right? At some point, you've sort of squeezed everything you can squeeze out of PDB, and methods in ML aren't super defensible over time, right? People will figure out the new tricks. People talk to each other. In terms of things that I'm excited about in the future, I do think, assuming a lot more high throughput data collection, we'll be able to learn a lot more about all the different epitopes and binding sites that exist on extracellular targets. We were talking a little bit about immunogenicity. Those are places where I think machine learning models want different data, right? Like, so library on library methods, sort of very high throughput types of methods. I think you could learn from those. So I'm mostly looking at the data collection space more than the individual modeling techniques space. So we'll see. I'm confident that we're still in sort of GPT 0.5, right? There's still a lot to be done. Where the exact thing is going to come from, TBD in terms of tricks. I do think data is going to make a significant difference. It's one of the reasons why we're also investing in it heavily.
Fantastic. Steph, with that being said, there are some more questions. Everyone, we're going to write a follow-up piece on this. We're going to release the recording of this as well. So we're going to try to answer many more questions in those materials as well. Thank you for joining. Thank you for participating. Thank you, Steph, for presenting. And we're looking forward to seeing all of you again via our email newsletter, via our blog, in our content, and at the events. Conference season's coming up, so hope to see some of you there as well. And with that, I wish you a good afternoon, a good morning if you're in the US, and we'll see you soon. Thanks, Steph.